And as you all are finding your seats, so as everyone in the audience is thinking about your provocative questions from the last 4 talks that we've had, we do have some charge questions to get us started. They're up there for me. They're not on the slide. We have some charge questions that I'm going to read to you because you know I'm going to go off script. I have a charge question that I want to read to you to get us started. And so there are 3 of them. So just to start off, in the data sets you've used for your models, and a couple of you touched on this, how do you deal with data quality, the source data quality, biases, sources of uncertainty? And we know that that exists in all the data sets that we're using, but what are the techniques that you use to deal with them? And then how do you transparently communicate that?
Okay, so this is actually a really big problem for a lot of the models that we're working on. We pull data from a lot of different sources, and we're pulling classification and labeling data. So it's pretty common that we'll have like a compound that is called a skin sensitizer in one data set and a non-sensitizer in a different data set. Okay. So when we first built these models, we took a precautionary approach. And that's really driven by, wanting people to be comfortable that these models are going to be really sensitive and maybe giving up a little bit on specificity. It turns out that that's really not the best approach. Actually, I just checked because I was suspecting this question. And at least until recently, PubChem used to list that water was an acute oral hazard with some percent, right? And that's just because like water has these impurities in it and someone registered that in a different context, right? But if you're going to take a precautionary approach and you've got like a 0.1% group or maybe even like a 2% group saying that, and it's because of just some side effect, then you're really going to damage the data a lot, right? So it's actually these chemicals that are like getting different results all the time that are exactly the hard ones to model. So one approach is just to kind of take all of these different opinions and work that into your model, right? So if you've got a loss function or something that's trying to optimize on what the data sets say, then you're going to represent that data, as a sensitizer, as a non-sensitizer, with the percentage that it's given to you. So I would say that actually modeling that uncertainty is at least one approach.
So something else that, so I work with a lot of the same types of data sets as Tom, obviously, and nobody likes to hear this answer, but one of the answers is hours and hours and hours of manual curation. It's just the cold hard truth. We've spent a lot of time really trying to dig into the source data, and you have all these databases that you can sort of automatically curate and dump into a larger data set, but if you lose the provenance of that data, then you sometimes will propagate errors throughout multiple steps, and then those get propagated into your training sets for your models, for example. One really interesting thing that came out of the work that I presented that we didn't really expect was the power of these consensus models to actually override errors in the training set and in some of the external validation sets. So we've taken like the CATMOS model, the consensus acute oral tox model, and we're testing it on an external set of new data, relatively new data, unpublished up until now from ECHA, from the European Chemicals Agency, that they shared with us. And we were initially concerned because there's, as you might imagine, there's a small fraction. It's a very unevenly distributed data set, right? So thankfully for all of us, there's not a ton of very acutely toxic chemicals floating around. So there's, a small percentage of chemicals look like category 1 acute toxicants, which with LD50s, of less than 50 mg per kg. And the ones that were labeled as such in our external validation data, there were several that the models were getting totally wrong. They were predicting them as completely non-toxic. And so we were really concerned. Okay, this has an LD50 of 5 mg per kg per day, and our models or consensus models are predicting it as totally non-toxic. Well, we went and we started to dig into the data set, and we found out that it was a transcription error, that the LD50 of that chemical was actually 5 grams per kg per day, not 5 milligrams. And that was an insight that was provided to us by the computational model. The same as actually occurred, anecdotally for chemicals that were in the training set even. So if you have a chemical in the training set that is misannotated, but if you have enough other chemicals that are similar to that chemical that are correctly annotated, then the overall, model will actually basically override that error in the training data because of the similarity and the persistence of other chemicals in that training set, and it'll compensate for that. So that's sort of one. There's sort of two ways. There's the manual curation approach, and then there's the sort of the ability for large enough data sets and consensus models to kind of override those errors in the data. And then the last thing I'll say about that is that we all, when dealing with regulators and trying to predict endpoints like classification and labeling schemas, we have to all be very cognizant of the fact that those are essentially arbitrary lines drawn in the sand. So, an LD50 of 60 mg per kg per day is really biologically no different than an LD50 of 40 mg per kg per day, but they have very different consequences in the classification and labeling space, in the personal protective equipment that should be worn, in financially, they have a lot of different consequences, and Reza could probably speak to that much better than I could. But so I think, as Tom said, being transparent about sort of the quantitative uncertainty or the quantitative confidence around the predictions, and communicating that maybe a borderline chemical is truly borderline, that there's a 55% chance that it's a category 2 and there's a 45% chance that it's a category 1, instead of just putting it in one box and saying that's the answer. That's one way to deal with it.
Very interesting.
The only thing I would add to that with respect to the omics data types, as I mentioned, the missing data problem is a big deal for us. And so the way that we approach that is we do use standardized pipelines for the processing, the normalization, statistical analysis that we do on the data sets to get to the sets of features that we deem to be important that we would put into the models. And we have well-documented processes we use for that. And then we also try to apply statistically driven imputation approaches to understand what's the level of missingness for which we can impute values and not change the structure of the data too much, and often can find a threshold for which more than 70% missing data and the features simply not useful, and so then we do have to do some data trimming to have increased certainty.
So for mixtures in environmental epi studies, we are basically seeing the same forms of biases, expecting the same forms of biases as in any epi analysis. We're expecting confounding, potentially selection bias, potentially, not potentially, most likely exposure measurement error. These are most likely going to be extremely amplified in huge data sets, especially if for example, measurement error is correlated across the chemicals that I'm including in my mixture. There are now some studies showing that we might want to include multiple chemicals in the study for co-chemical confounding, but that could also induce amplification bias if I have confounding by other factors, so we should also be worried about that. These issues are not even completely resolved in a single chemical analysis. There are methods still being developed. And understanding how this would impact my analysis are likely going to be specific to each analysis. So I think there is a lot of work ahead of us in understanding and developing methods to deal with these things. We have a hard road ahead of us, but we are trying, hopefully people are trying to be very explicit. And it's not so much in the community because we more or less know about this. So yes, if someone does not include exposure measurement in their discussion, I would expect that there is. But it's more for dissemination of these findings in other communities that is extremely important. Yes.
Yeah, very helpful perspectives. Maybe we could turn it over here for a question. And if you could please identify yourself and your affiliation.
I'm Risha Putzrath with the Navy and Marine Corps Public Health Center. My comments are my own, not those of the Navy. Sorry, we have to say these things. There are two issues that I haven't heard addressed that I was wondering if anyone is exploring in the area of machine learning. One is when dealing with well, not just mixtures, but any interactions. Interactions can change with exposure level, for example, going from independence of action at low levels to antagonism at higher levels. So you're not dealing with one interaction, but a change in the basic equation. The other is in dealing with non-chemical stressors and the interest that we all have in how to combine them. And I at least haven't found a systematic way in the three areas I've dealt with to combine chemicals in milligrams per kilogram per day with radiation in grays and pathogens in either colony-forming units or plaque-forming units. So I was wondering if anyone has been addressing those issues.
I can start, and people can jump in if they want. So for the first part of your question about the complicated nonlinear interactions, actually machine learning is a great way to help us deal with this. Many machine learning algorithms are very flexible. They can help us look at nonlinear potential synergisms in this high-dimensional space. The concern with that is that some methods might tend to be too flexible, so they might be too susceptible to outlying and extreme or influential values, but they're definitely allowing us to better assess and capture these complicated phenomena way better than, for example, a linear regression with just a penalized spline. So I'm very optimistic about adopting machine learning methods to look at these complicated data structures. For chemical and non-chemical stressors, or even chemical stressors but at different units, that really depends on how we want to combine and look at these things. So if the interest lies in creating one index of exposure to all of these, then maybe some form of standardization so we can pull things together might be necessary. It depends on the method, but not necessarily always. We could look at them in a hierarchical structure, so we can have a hierarchy on, sorry, I forget the three things that you mentioned, but one was radiation. Let's say I'm going to say socioeconomic status, and I'm going to say air pollution just. So we can put some hierarchy and allow both these latent groups, assess effects of these latent groups and potential interaction of the latent groups, and then also of their members underneath, both within and across groups. This is just off the top of my head, one potential way that you could go forward, but there are ways to deal with that. I don't think units is so much the issue. The issue is if the scale is very different, because the model, the statistical model, would not know what the units are. But if the scales are very different, then that might mess your model and its convergence. So you might need to standardize just to facilitate computation. I don't know if that answers the question at all.
It does partially. If I may throw in one more confounder. With things like chemicals and pathogens, there's sequence and timing of exposures as well as amount. And I'm just wondering if people are addressing that.
Thinking of that, yes. Addressing it, I'm not sure yet. Trying to would be there, yeah.
Yeah.
Katrina,
So one way to think about that is if you characterize it as a pre-existing, exposure versus, and the subsequent exposure, you could code that in a binary way. For example, in a model where you could say, did these folks have this, exposure prior to exposure to this, and then include that in the model as, just for the sake of determining, is there an interaction between those things? And then if there is, pull that apart and start pulling the string to build a more detailed model.
I also, to add to that, I think going to something that Katrina mentioned at the end of her talk, I think this is where, the promise of wearable biosensors really, has a really exciting future, because what you really need to start answering that question and developing appropriate algorithms is Timecourse Data. So, to be able to appropriately transform your variables such that they can be combined using multivariate approaches, you need to understand the relationship on both, on many scales, the most important of which really being the time scale.
Thank you.
Yeah, just my 2 cents on it. My world is not mixtures, so all of these answers are a lot more informed than what I'll give you. But the problem with my approach, taking this structural data and trying to predict an outcome, when you bring it to mixtures, is the combinatorial problem, obviously. And then if it's going to be continuous, it's just a complete no-go. So if you want to apply it, it's probably you have to go directly to this biomarker genetic type data, right? And then it depends what your context is. If you're going to say I need to have a model that predicts for all of these different combinations before I'm exposed to the mixtures, that's going to be extremely difficult. If it's someone was exposed to something, I don't need to know what it was, but I can take some biomarkers from that person. I mean, already people have amazing models that are built just directly off of biomarker data, for example, for tracking patient survival in cancer. So I would back up into that kind of data.
And that's a really nice segue into the next question, which is, this workshop is put together by the Standing Committee for Emerging Science for Environmental Health Decisions. And that environmental health decisions, the so what component of it, is what we're also trying to focus on here. So as you're thinking about the algorithms that you've developed and the work that you're doing in your field, how and by what mechanisms have you brought in decision makers, the people who need to deal with a wide range of stakeholders and perhaps different stakeholders in making those decisions? And can you share with us, when you brought in those decision makers, how did that change or influence the approach that you were taking, the communication you had? Can you share your experiences for the group?
I'll start because I work with a lot of them. So, that's the entire purpose of ICVAM. So ICVAM is the Interagency Coordinating Committee on the Validation of Alternative Methods. And the roadmap that I, was promoting during my talk was really a function of multiple meetings where we brought together regulatory decision makers, industry stakeholders, NGO representatives, and basic scientists to start to build a framework and understand how is the best way to go about tackling something as difficult as modernizing our approach to chemical and medical product safety testing. And I can tell you that the most valuable thing that came out of that entire exercise was the necessity, the absolute necessity, to have the end users be participants in that process from the very beginning. And sometimes it's a little frustrating because, like I said, sometimes you're confined by, semi-arbitrary classification and labeling schemas that they would like you to predict. And that's not as scientifically fun as predicting something like, complex biological mechanisms and diving deep into the signals of the biology. But ultimately, to be pragmatic and to have your work actually be put into practice, you have to understand what it is that regulatory decision makers are grappling with and what they have to deal with on a day-to-day basis. And so listening on the front end and then trying to help design tools to meet those needs is really what we took away from that.
And do you have any specific examples of something that maybe you did differently based upon that conversation than a preconceived notion that you or others may have had going into that engagement?
Yeah, I mean, the Catmus project is a perfect example. We asked them, okay, what are the endpoints that you use to make regulatory decisions when you get submission packages of data? How do you currently apply that to your exposure thresholds or your PPE requirements or your packaging requirements. And so those were the endpoints that we then asked the modelers to target, instead of, trying to say, okay, let's model all of the biological mechanisms of acute toxicity and really get at the pathways underpinning those endpoints. Let's do something practical and model the endpoints that are essentially what they would be getting out of the ultimate black box, which is the animal study itself.
So I would provide a couple of examples for us. So in all of our clinical interactions, we always work with the physician scientists right up front because we're analyzing their samples and they have their questions. But they're very heavily influencing the feature selection part of the models. They will look at the metabolomics data where we see, for example, big changes in one specific small molecule or another, and we'll say, oh, the carbonate ion seems to be really highly predictive, and they're just like, oh, that's really variable. Like, nobody will ever believe that. Clinician will never measure it. Let's move on. And so they're really, really essential at helping us narrow down the list of features that we're going to focus on for what we would pursue in the models, and also where we have the collinearity and correlations within the data that we see. They can help us determine from their experience in the clinic, which measurements do they see that always pop up together, and so we have redundancy in our data, and try to focus down on where are the unique predictive features. The other example really with the first responders and the military that we're working with is that question of, what is their priority? And their priority is to assess the health and the performance of the individual in the field. And yes, they do care about the development of chronic health conditions 20 years, 30 years later, and the VA is a big customer of those questions. But at the beginning of the day, they're interested in, do we need to get that soldier out of that environment in 24 hours, or they are not going to make it home? And so that has really helped us kind of narrow down the selections of features to what can we actually measure in a deployable device, and what kind of measurement technologies do we have for assays right now where we could say this set of small molecules can all be measured in a colorimetric assay and we could deploy it in the field tomorrow, as opposed to these other markers are going to require a lot of chemistry development, a lot of new assays that are maybe not field deployable today. And so that's really influenced our process.
I just want to add one more thing on to what I said. I realize it may have sounded like I was implying that the biological relevance of what we're predicting is not important, and that's definitely not the case. I think another really good example of where the field has really evolved in terms of incorporating the input of end users and biologists at the same time is with adverse outcome pathway development and the whole concept of the AOP framework, where if you can actually map the progression of toxicity from molecular initiating event through key cellular events down to adversity, and then you can build integrated testing strategies that cover those key events along that AOP, that actually really helps provide confidence in the minds of the end users and of the regulators that are trying to apply those. And that is the case even if it's a more complex machine learning model that they may not understand all of the nuts and bolts of. If they can see that the features that are informing that model are biologically relevant from sort of the mechanistic pathway standpoint, then that really is an added benefit.
Yeah, it's explainable models, explainable inference that Katrina was talking about.
For the mixture side, I think EPA has recognized the necessity to think about regulating mixtures, at least in the air pollution realm, in which we have been historically, I guess, spoiled in that the Clean Air Act is requiring EPA to revisit all new science every 4 or 5 years and adjust standards accordingly. Things potentially have started to change, so we might need to rethink how we reach out and include more decision making to make sure that our work is being heard. It is challenging, though, to think of how we could regulate mixtures, because they will be very different from, say, again, air pollution from city to city. So that is an added challenge, and we do need to start thinking more about this.
So it's a pretty mundane side is just by talking to a lot of the companies that are starting to use these tools, learning how they're using it. There's a lot of just engineering a tool. You know, it's like, well, now I have to build a programming interface. Now I have to build an Excel export. I mean, that's really basic, right? And people say we need that to do that thing, and you have to build that. Maybe a more interesting side of it is when companies start using these tools, they come in with their own attention mechanism, right? These are the chemicals I care about. And when they tell us that, those are going to be areas that we focus on because it becomes more important to building that tool. So I'd just say the attention mechanism is the biggest part I see playing in terms of the actual model development. This is stuff I care about, therefore I'll make the model better at that stuff.
So we have a question on this side of the room. If you could give your name and affiliation, please.
Hi, I'm Alex London from Carnegie Mellon University. I appreciate everybody's talks. In a lot of the talks and in some of the comments, I've heard discussion of mechanism. I don't have a well-formed question, but it seems like there's a bit of a tension between large-scale methods that leverage correlations in data sets and then this talk about finding, leveraging, explaining mechanism. And so I'm wondering if you could just maybe each say a little bit more about what you mean by that, the degree to which that mechanistic knowledge is brought in from outside as prior constraints on what you're doing, and in particular the degree to which the particular machine learning methods that you're using are shedding light on mechanism, or whether you didn't intend to imply that.
So the way that we think about the mechanism, and it's for the omics data, we do a lot of enrichment analysis to pathway databases that exist to map these data types. For some of them, like lipids, there are lipid maps. But of course, if you map lipid data to gene ontology, you're going to get lipid pathways, right? I mean, it's pretty obvious. But the way that we tried to use that is it's a very iterative process with the machine learning where we can use the machine learning in an unsupervised way to identify features that seem important. And then we can do some clustering with the features to figure out those that have the collinearity, those that exist in the same pathways, and then collapse some of those features down into like feature categories where we lump them all together into a category. And that can help us see where one pathway plus another pathway plus another pathway increases your predictivity, where you really maybe just need to measure one component of each pathway as opposed to all measures of each pathway. Where it's important for us is in thinking about, with the application for the clinicians, and when we start thinking about mitigation approaches, how are we going to target this pathway in order to mitigate the problem, to prevent the problem from like a drug targeting perspective? And so what we're going to do is want to know these pathways so that we can see where are drugs that already exist, that we could repurpose medications that are already out there, or where are there new drug targets where we see the opportunity to develop something that's going to prevent or mitigate the action.
So I think it's an excellent question, and it's a problem that we grapple with all the time. I think there's a constant balancing act between having predictive models that are successful because they have large enough training sets, they cover a diversity of chemical space, and therefore a wide range of mechanisms. The acute tox is a great example. There are many different ways to die, right? All 15,000 chemicals in our training set are certainly not acting through the same, lethality mechanism. But in that case, we probably have enough coverage of the chemical diversity that these QSAR models can be successful. That being said, that for a regulator to have scientific confidence in applying those models, sometimes there needs to be the ability to have mechanistic interpretation. And so, random forest is one of my favorite machine learning techniques, simply because it's so easy to extract variable importance scores. So you can, as I think it was Jason talked about this morning, you can go through and you can actually randomize each of those variables, and you can see what the effect it is on the overall predictive performance of the model for each of your individual features. And that is something that I've found very, very useful in communicating to regulators and to end users. If I can say, okay, here I have this machine learning model, and I don't need you to understand all of the math behind how it works, but here is a variable importance projection, and here are the top 10 features that are informing this model. And if they look at those features and those are biologically or chemically meaningful, then that really helps give them confidence that this is something that that is scientifically defensible, or even in the case of the EPA, legally defensible.
This is a very good question indeed, and I think this is not only related to machine learning. This has been an issue in epidemiology in general, and what most people tend to do is incorporate this prior knowledge on pathways implicitly in the model. For example, if I was going to run PCA, which I don't know if it's technically still considered machine learning because it's been around for about 100 years, but then the number of components that I choose to use in the model is based on yes, there are some criteria that could be used as guidelines, but there's a huge subjectivity to it. And this can only be amplified as the methods become more and more advanced. So I think we should try to develop, extend methods to incorporate explicit incorporation of this prior knowledge, that we can test the sensitivity to it. How much do our results depend on what I am thinking that I am leveraging from prior knowledge?
So for me, there are at least two big parts. One of them is like spurious correlation. Like when you've got these big data sets, you kind of assume that's going to drop out. It's not the case. So like if, for example, if you're collecting classification and labeling data, sorry, I keep bringing it back to it, it's what I focus on, but yeah, I mean, there can be situations where a test is only performed if something else has happened, right? And so that can create all sorts of weird sampling problems. So one way to combat that is let's get lots of different data sources, really different, right? So we can kind of combat it a little bit that way. And then I think there are a lot of other approaches that I'm not going to get to in 8 minutes, but the spurious correlation aspect is like one aspect of why mechanism is important, because we can try to get rid of that, right? But it's not easy. Like, it's not always easy to just say, like, that's obviously a spurious correlation. And sometimes you have these spurious correlations that are subtle and, seem like they should be causative. The other part of it is just the counterfactual part. And I think it's really important to anchor that somewhere. So, like, if you're using, these models to predict something about a chemical structure, the question you're asking potentially from a mechanism perspective is like, what happens if I change that chemical structure? So that is not really what most people here, I think, would mean by mechanism, but I think it's a good place to anchor it, because you can say, maybe I can change something about the structure and organize my models around answering questions like that. I'm always going to answer with an engineering answer, but just to say, there are a lot of people working on causal models now too. It's a really interesting place to look in the literature.
So despite what that clock says, we only have 1 minute and 30 seconds left, 1 minute and 29 seconds left. And as such, if Patrick wants to ask a really quick question that only one person provides an answer to.
I'll talk quickly, and we can just deflect until later if we need to. We were just talking about the need to listen to the stakeholders that we're producing these things for. Very laudable. Are there challenges in the other side of that, communicating to them how they should be thinking about these models, and maybe how they need to be nudged to think differently about their problems, because we can answer different questions with the new tools?
Well, there's a very easy answer to that, which is yes.
Yes.
And that's the end of this session. Thank you very much. For session number 3, we're going to have Melissa Perry take us to the next section.